Good morning. Today we'll be focusing on the cancer research, and the outline for today's talk will be the optimization and automation of your dissociation, so any solid tumor tissue that you will have, how to dissociate that properly to then have an optimized downstream analysis. Further on, I will go into the isolation and also the cultivation of tumor cells, something brand new that we offer, a cultivation of primary tumor cells. Also then the TIL isolation analysis. Here we offer a new technology which is called the release. So you're actually able to get rid of the labeling that you put onto the bead on your cells, so that afterwards you have a label-free cell again. And then we'll finally finish with the isolation of tumor cells from FFPE samples. So we're able to isolate them now into single-cell suspension and then actually sort for them. So now today we are talking about a tumor microenvironment. We don't focus on the cancer cells themselves anymore. We actually go into the tumor cell and tumor itself and look at all the players that are involved. Here just a rough overview of the players that are involved, and just to give you an idea of how complex this environment is getting now. How do we dissociate this and get into the proper analysis downstream? Well, we sort of work with this workflow. I will go in basically chronologically through my talk with this, starting off with solid tissue, then going into the sample preparation is what we call when we dissociate the tumors, and then going into the cell isolation to basically enrich for your target cells, and then any downstream analysis that you might be interested in. So what kind of issues might you have when you go along with this workflow? Well, the first part is when you dissociate, you might degrade your epitopes. So this is a very crucial part where you really have to be careful to not degrade these, because these will be needed for any downstream sorting or isolation that you might do. Also, therefore, you will not be able to recover subpopulations because you're not able to identify these epitopes. This will also be worse if you are not able to reproducibly dissociate your tissue. So this is why we have a solution also to do this in a very standardized way. Afterwards, when you have single-cell suspension, you want to isolate so that you don't have low frequencies of your target cells, because if your target cell is a rare cell population, you will have difficulties to do proper analysis with them. Also, we offer automation possibilities to, again, standardize your procedure. Long acquisition times might be a problem for you if you have this. If you are looking for these rare cells within your tissue, then you are going to be standing a long time at the flow cytometer. Also, you might have your problems with background staining. So this comes, of course, through some FC region binding or other specific binding, and low reproducibility. So all of these issues I will address during this talk, and where we offer solutions to address these problems. The first problem you might actually have is just that us in Miltenyi, we do not have a hospital located right next to us. So we need our tissue shipped to us, and this means that it's at least for 24 hours on the road. This is not good for the tissue. It's going to start to have some apoptotic or necrosis happening. It will degrade. So what we did in-house is that we actually created a media which is for storage of this tissue. And after a while, we noticed that our customers would also be interested in this, and we made a product out of it. Just to be sure, we also compared it. We compared it to these two, as we call them, compared to SNC. These are basically organ transplant solutions. These solutions are GMP and are very expensive. And ours is research use only, and it's at a rather cheap price. So basically, it's a great way to give you the flexibility of when you start your assay. So normally, the surgery of a patient will be in the morning, and you will get your tissue in the afternoon. This means if you get the tissue and you want it fresh, you're going to start your experiments in the afternoon and be in the lab until late at night. This way, you can just store it. We guarantee 48 hours. This is the time frame where you'll be sure that there's nothing going to be happening to your tissue. Anything further than that, we've had it in also up to 5 days, but it depends on your tumor tissue. If you are working, for example, with pancreas, there's a lot of enzymes involved. You will see degradation starting off quicker. But this 48-hour period is what we guarantee that nothing happens, and gives you the flexibility to start your experiment when you want to. So when you do have your tissue ready to go, as I mentioned before, you want to dissociate it. It's a crucial part that you want to take all cells apart, but of course you don't want to destroy them. And the other point is that you do want to preserve the epitopes. So to guarantee this, what we offer is a combination of mechanical plus enzymatic digestion. This is all done on this instrument, which is called the GentleMax. What it does basically is that it has a special tube that has a rotator inside, which will then do the mechanical dissociation. And on the other side, we have enzyme kits, which are purified enzymes. So why do we use purified enzymes? Because this way we control which epitopes, or at least which strength of the enzymes we are dealing with. So if you're doing a normal overnight digest with a collagenase, each collagenase will have side activities, which is good because you need some side activities to actually dissociate your tissue properly. But you can't control it because each lot of collagenase will have different side activities. So if you have a tryptic side activity one time of 3 to 4%, and next time it's 5%, each time you need to then start over seeing how much collagenase they need to use to get the same dissociation that I had before. In this case, we have purified enzymes, 3 of them, so this way we control which activities we have. And there's no side activities because they're purified, and we need to have these 3, which are not as harsh, but therefore combining them with the mechanical, we still get a very nice single cell suspension of viable cells. So this is the key to this instrument, that you basically combine this mechanical with enzymatic digestion, and therefore have a very gentle way to get your cells in single cell suspension. This is just one example afterwards. So you have some of the cells here. We gated out the red blood cells, and basically we see that we get a nice viability, and also the yield is good. We also plated these cells. You can see here after 2 and 10 days, and they grew very nicely. One of our collaborators, so David Gillam at the time of the University of Manchester, he also tested his version, so his overnight digest with collagenase, against our Gentlemax dissociation. And here for the CD4 and CD8 markers, he tested 4 different patient materials, and he saw that he gets the 2 peaks, which is the filled lines, when he used our Gentlemax dissociation. And when he used this overnight digest, he only sees 1, which is the empty line. This indicated to him that some degradation had taken place, because now he's only seeing 1 smear instead of the 2 high and low markers for these epitopes. So basically, he was being reassured that our version was the gentler to his cells and not losing any of his markers. Also, he took it a step further because he wanted to see, are my cells still functional once I dissociate with this Gentlemax? So are they still happy, basically? And what you see here, he tested, he was searching for TILs, and what he did was he tested for IF and gamma release, and this top row here shows when you digested overnight. So from all these patient samples, he only got 2 that actually showed some nice IF gamma release. When he tested with ours, all of them showed some release. And when he compared them to each other, always our Gentlemax showed a higher IF and gamma release than his, basically. So this showed him that the cells not only had their epitopes intact, so that he was able to recover them in his assays, but he also showed them that they were functional, fully functional, so that they were not stressed. And I was mentioning before, we offer these. You don't need to read these in paste. I just wanted to show you that these are available online. What they are basically is a long list of 400 epitopes that we both have for mouse and human that we tested for you, so that when you start your experiment, you actually know in advance which epitopes will be attacked and which won't. So this gives you the possibility to already play around with the enzymes in the kit that we have. So there's basically the 3 enzymes. They come separate from each other. And if you see that, for example, in here, CD25 is, for example, a sensitive marker. CD25 for Tregs, very sensitive. If you want to keep that, you reduce enzyme R to about 20%, and all of a sudden you have all your epitopes stable. The only differences that you will have is that you won't be able to dissociate quite as nicely as before. This is why we have enzyme R fully in there normally. But since you are looking for a Treg, it's a TIL, which is the first cells that come out. So you will not have any loss in that, but some of your tumor cells won't be dissociated as well. So nicely. That's the only difference. Also, if you are using trypsin, over 50% of the markers that we tested are affected by this. So it's a really harsh enzyme. You should really be careful with that, yes. And we actually only have about 5% of the epitopes that are somehow affected by the kits. One of our R&D persons also questioned us then, so if the enzymes are so bad to lose your epitopes, why don't we leave them out and just do it mechanical? Then you wouldn't lose any of the epitopes. So what he did was he tested it with the enzymatic digestion, and you see for PD1, LAG3, and so on, you see these populations popping up quite nicely. Now he left the same sample, no enzymatic digestion. And you see that you lose these populations. Now, why do you lose these populations? It's not because the mechanical dissociation destroyed them, somehow broke open the cells or something. No, it's because the cells are still attached to other cells because they did not release. So basically you are gating them out as doublets or something else, but you're not catching them as their actual population of PD1 or LAG3 in this case. So you'll be missing these populations because you're not properly dissociating them from the rest. So it's crucial to use enzymes. So now that you have a single cell suspension of hopefully very viable cells, we go into the isolation. Miltenyi, of course, famous for its MAX technology. What we do, we label our cells, our target cells, with an antibody that is connected to a bead. And this then goes onto a column, which is placed into a magnet. Everything that is labeled will stay on the magnet, on the column, and everything else goes through. Once you take the column out of the magnet, you elute the rest, and you have separated your cell populations. This is the standard procedure, and this is how we can play in different ways with this. And what we're going to do now is we're going to do this for tumor cell isolation. So if you want to isolate tumor cells from your tissue, just the tumor cells. So we have 3 different ways to do this, either coming from a PDX model, coming from a mouse tumor, or coming from a human tumor. It's always the same workflow. You start with your tissue, you dissociate, you have single cells, and then we go into isolation. The only thing that changes are the reagents because of the different tissue types. Excuse me. So why do we think this is important to do? What we did was we did a little test. So we took a tumor cell line, we injected into a mouse, right and left flank. Same amount of cells, same tumor line, and we injected it into right and left flank of the same mouse. So now we let it grow, we take out the tumor, and we compare the 2 tumors to each other. And usually we'd expect that these are the same tumors. So everything is equal to each other, but it's not, because the infiltration rate varies on a high level. What we see here in these 3 samples, this is the most extreme, of course, that this one got much more infiltrate than the other side. So this means if you were to compare these 2 tumors to each other, you would be getting a large bias because one was infiltrated with much more cells, infiltrating cells, than the other one. So you're not comparing the same thing to each other. So this is why we thought we want to get you to a possibility that you can only compare tumor cells with tumor cells and get rid of everything else. And this is why we have this mouse cell depletion kit, in this case for the PDX models. So what we do is we label with a cocktail of these antibodies that are labeled with beads 2 target cells that are all mouse-specific. So this means that you're pulling out everything that's mouse-specific, and your tumor cells, which are engrafted, will go through, meaning you have untouched tumor cells, pure population, to do any kind of downstream assay. To go for a second isolation of specific subpopulation of these tumor cells, you don't even need to know the marker on your tumor cell, because everything that's non-tumor will be detected, because we're working with 2 species, mouse and human. Everything that's mouse goes out, everything that was engrafted goes through. This is the great advantage of this. So here you actually have the flow data also showing here, where we have the original fraction, the negative fraction with EpCAM positive cells, and here then your positive fraction, which are basically the mouse cells. So it's a very simple procedure, 20 minutes in time, and you have a pure tumor cell population instead of working with all these different cells that are going to be interfering with your analysis downstream. Of course, we've tested this on multiple tissues. So here we have renal, lung, and also bladder cancer. Again, you have the original fraction where you see that all cells are still present, and then your isolated EpCAM-positive tumor cells. This is what it looks like when you go through the microscope. Again, we stained Vimetin with red. We have the EpCAM in green, and also the merged picture would look like this. So this is your starting material of your single-cell suspension, and when you isolate, you get your pure tumor cells. So basically, you're not getting a bias from everything else that's in there. One of our other collaborators, Jeremy Rich at the Cleveland Clinic, he also made a nice discovery when he was beta testing this for us. What he did was, he's working with glioblastoma. So glioblastoma has a lot of dead cells, debris, the myelin sheets, so a lot of junk that he's not interested in. And when he has dissociated this, he normally got a very low amount of viable cells. So in this case, about 9%. And out of those, about 75% were tumor cells. So his issue was that he wanted to have this as a cleaner sample, and when he used the mouse cell depletion kit, he was able to clean this up to 98% cells, so viable cells, and out of those, 98% were also tumor cells. So this is great, efficient debris removal in that case as well. So not only are you purifying your tumor cells, which you want to work with, but you're also at the same time getting rid of everything that you're not interested in, as in debris, which would be just clogging up your instrument maybe after downstream. What we also tested was spiking in 1,000 labeled cells into a 1 ml of blood, so mouse blood. And when you look at this in the flow cytometer, what you see is that you see nothing, because 1,000 cells is just too little to identify in 1 ml of blood. So what we do is we do a red blood cell lysis, and you start seeing these 3 dots here. I'm sure that nobody will recognize this as a proper population of cells, but when you do a red blood cell lysis and add the mouse cell depletion kit, you're starting to see these cells over here again. And this is what you were looking for. And in this experiment, we're actually able to recover 803 of the 1,000 cells that we spiked in. So that's a pretty good recovery on these cells that we basically tested in this experiment. So it works also on blood. And another collaborator from us, Massimo Moro in Italy, he's looking for disseminated human tumor cells in the PDX model. And his issue was that these disseminated tumor cells are in such a rare cell number that they are at 3 to the power of -4, and this really makes it hard for him to identify them and to work with them. But using the mouse cell depletion kit, he's now able to purify this, or to amplify basically these cells to a 0.25%, which is a huge increase for him, and therefore makes it easier for him to work with. Another advantage is also that showed John Dick in Toronto, who is actually using the kit off-label because he's not using it for tumor itself, because he's engrafting, he's looking for HSCs. But as I mentioned, since it's mouse and human, anything you engraft will be isolated from all the mouse cells. So basically you can use it in that way as well. And the great advantage for him was that he was getting, using this mouse cell depletion kit, homogeneous starting material. So every time it's the same, instead of the beginning of the black lines where he has like varying amounts of HSCs that he was starting off with. So basically he was leveling all his experiments to the same level, which was a great advantage. Also, we have posters, scientific posters available that we do show at conferences. If you have questions, do address us, and we can send you these posters to explain further. I will skip the mouse part due to time reasons, but it's the same principle as with the mouse cell depletion kit. So now that we're working with mouse tumor only, it would be all the non-tumor mouse cells being attached, and everything that's mouse tumor goes through. And this is the same for the human one. Here I do have some extra data, so I will go into it. You see in the original fraction, you have your fibroblasts are still in there, your endothelial cells, your blood cells, everything is still there. But when you then do the isolation, so again, it's a 20-minute procedure, everything that's non-tumor will stay on the column, all the tumor cells go through. Very basic 20-minute procedure. Again, in the microscope, you would have here your vimentin and your EpCAM cells, so your cancer cells, and then afterwards, a clean population of your tumor cells. Also, it works on quite difficult tissue. So here we tested pleural effusion. Basically, again, you have on the top row the original fraction, and then on the lower row the isolated tumor cells. So this just shows that it was for ovarian carcinoma, pleural effusion, that it also works. It's very sensitive to kids, so it works on these difficult tissues as well. Here, just an example to show you, to compare basically the original fraction. This is the single-cell suspension directly after dissociation. So coming out of our Gentlemax, you see something like this. You wash it 5 times, you will still have a very heterogeneous population of cells in there, and if that's what you're going to work with, you're going to have a lot of readouts from different types of cells. But if you now do this tumor cell isolation without any wash, this is just an amplification here, you see that you have a very nice population of tumor cells. This again is ovarian carcinoma with the double nuclei, which is a hallmark for that cancer, which gives you a nice population to start off with. Also, very important if you go into NGS downstream and you want to do any kind of sequencing there, it's the same procedure again. You have your tissue, you dissociate the single cells, and now you isolate for tumor cells. And this then goes into exome sequencing. What is the advantage here? Well, if you see here, the blue circle would indicate what you do if you do not isolate. So you would get these readouts in the middle, which are basically the ones that you're interested in. But you get additional readouts that you somehow need to calculate out because they're false readouts. Especially if you're working with PDX models, you will get a lot of mouse reads that you need to calculate out. If you do the isolation, you don't get any of these mouse reads or any of these unwanted reads. You get your normal reads, but you get an additional, significant amount of additional reads because you're increasing the sensitivity of the reads, therefore also the quality downstream. Because you've removed all that's not interesting for you, You're increasing the sensitivity of the sequencing, basically. This is the same, basically, showing it in a graph where we have an increase of the frequencies that you get actually in comparison. So this is the isolated tumor cells and the bulk tumor cells. What I actually want to point out is these two examples still, where we're working with the thymoma here. So if you have NGS data, you might be able to interpret something in a way or another depending on what you see. In this case, and if you had just done the bulk tumor, you would see no mutation at all. But now looking at the actual isolated tumor cells, you see that a mutation does pop up. So you would have missed this mutation had you not done the isolation. Also, in this case over here, you might identify this as a heterozygous mutation, as shown in the bulk tumor. But when you look at the isolated part, you actually see it's a homozygous mutation. So these are little errors that you might read into your essays because of working with so many other cells that are included in there and that give you a wrong or a biased readout. Again, if you are interested in the NGS topic, we have scientific posters. Do approach us. We can have them sent to you. So now that we've dissociated tumor cells, we have them in single suspension, we now isolated tumor cells, and we can go to the next step and we can actually cultivate these tumor cells. So those primary tumor cells can be cultivated into stable cell lines. At the moment, we have this for pancreas tumor only, but what you can see here is that they grow both in adherent cell lines and suspension cell lines. They also work as unsorted or isolated tumor cells. Why am I saying this? Because what we do in our own labs is when we're testing to get this tumor media ready, is that we take a tumor piece that we get, we cut off a little piece, and we place this into a well to have it grow, to see if this grows out, like an organoid, basically. What we do then is we take the rest, we dissociate it into single cells, and we take a part of that and just put it also in a well, but it's including all the fibroblasts and TILs that are in there, having the whole mass. And the third part is that we take the rest and we isolate the tumor cells and put those to grow. And then we have 3 samples where we grow tumor cells and we see which one grows best. We cannot say in advance which will be the one that picks up and which will expand best, but this is the best option to see, because even in pancreas, different tumor lines will have different ways of picking up in the best way. This is what our experience shows. This has been released now in October, so it's very new. And we will also have a guideline coming out which will give you these tips and tricks where you show when to change media, how to get rid of fibroblasts. We should add a ROCK inhibitor, all these kind of things that we've learned while we were doing this, how to best basically start growing your tumor cell line. At the moment, it's for pancreas, as I mentioned only, but we will expand it to other tissue types, such as renal and ovarian that are coming out, and then breast cancer also later on. So if you're interested in that, do contact us. This is an in-licensed product from the Andreas Trump Group in the DKFZ in Heidelberg. So there's also publication available on this from Nature that you can have a look at. Also, there's the great advantages that we preserve the heterogeneity of the tumor. So this is very, very crucial. We are actually now able to have a tumor that compares to the initial tumor that you had, and not, which I will get into, a tumor cell line. So this is a nice experiment that we performed where you see that we have patient material here from 4 patients. We took that tumor, we engrafted it into a PDX model, let it grow. We then took it out, grew it about 15 to 20 passages in our cell culture, and then we took it again and engrafted it in another mouse. And if you see, this tumor still resembles the initial tumors that we had before that. So we're really working with the same at least morphological, from a morphological point of view, with the same type of tumor. This is not the case if you're working with cell lines. So PANC1 is one of the most famous cell lines that is available for pancreatic tumors, and this one has been around for decades, so it's completely out-differentiated. So it will not show this morphological picture, basically, that you see when you're working with initial pancreas tumors coming freshly from patients. So you lose the heterogeneity here, and in this case, if you're working with these and put these into fresh cultures, you will improve and have a higher predictive power in your model. So it's really crucial to have a tumor that compares to the initial tumor that you had. Also, if you might think that we only checked on a morphological level, we also went to the genetic level. Does anything change on that level? And what you see here, we compared early passages to late passages, and the correlation of coefficient here, the ideal is to have it on a line, and you can see that this nicely fits. Also, if you compare the xenograft tumor versus the initial patient tumor, we see again the correlation coefficient demonstrates that this is genetically stable. So therefore, we can also say that not only on the morphological side, but also on the genetic side, the tumors resemble the initial tumor that we had first in the beginning, coming from the patient directly. This is the media. There's always going to be one basal media, which will be the same. And then the supplement, which comes over here, will then adapt accordingly to the different tumor type to see which tumor type needs what kind of extras, basically. So this allows for efficient derivation and expansion of tumor cells. We also preserve the heterogeneity, also tumor-initiating capacity, what we tested, and then also the genetic stability. Also, this, of course, then improves your model in vivo, but also in vitro. And you're able to create these stable cell lines that you can work with and do lots of assays, drug screenings, and so on. So this is really crucial. Now that we have these tumor cells and we can play around with these tumor cells, we'd also like to have a look at the TILs, so everything that infiltrates into tumor cells. So now we're switching from whatever was tumor to everything that is non-tumor, that is infiltrating as what we call TIL, which is CD45 positive. Again, it's the same procedure. We have an antibody to a bead, except at this time, we are labeling the cells of interest. So this time, our target cells are here and not the tumor cells that went through. And then afterwards, when we take them out, we have our CD45 positive cells. Again, here in the dot plots shown again. So why do we think this is also important to enrich for these? Well, if you have your tumor and you've dissociated them into single-cell suspension, this is just a rough image of what it might look like. You have a lot of tumor cells and some of these TILs that are infiltrating here. Now, if you try to do your analysis on this, you will have difficulties identifying your populations because they're rather small in comparison to all these bulk tumor cells that you're working with. If you take the same sample and you get rid of all the tumor cells and only look at the TILs, all of a sudden these populations start popping up and you will have a much easier time identifying your populations and be able to work with them, and even identifying the subpopulation of the subpopulation, basically, which you would not be able to do in the other event. So just to give you an idea, at the moment we have CD45 microbeads for TILs in human and mouse, and CD4, 8, and 4 and 8, so what we call a pan T cell, basically isolation for mouse. These ones will also come out for human now in December, but then they will already have the new release technology that we are working on now, so which I will go into in a minute. So just that you have an idea, if you can either go for all TILs or basically T cell specific. Just to give you an idea what the effect is, if you do this, you don't need to read the exact gating that we did here, but basically what you can see, this is before isolation, 100,000 events on your flow cytometer. And this is the same 100,000 events after isolation. So before isolation and then after isolation. Each time exactly 100,000 events. So it just facilitates and makes your whole analysis quicker. You're able to identify them quicker, you're able to work with them better. That's the point why you should do this enrichment. And it's a 20-minute procedure. And of course, the rarer your cell, the bigger the effect you will see. So if you're working with CD4, for example, positive cells, you see that you started off with 0.19% within this tumor. And just by doing this 20-minute procedure, you get 90% enrichment, so an enrichment to 90%. So this is a huge increase because now you're looking at very rare cell. Of course, this also works with CD8s and the pan T cells. And the purities, when you use these kits, are always above 80%. And as I mentioned, it can be up to 500-fold enrichment. But then again, this is depending on how much of your target cell was in the initial population. And the rarer it was, the bigger effect you will see. Also, if you might be worried that, but now we're actually touching your cell, so there's actually a labeling on your cell, does that affect my cell in any kind of way? No, it does not. So what we have shown here is that these activation markers, they don't react at all. So we can really guarantee that your cell is not activated in that sense at all. Also, since we are using the columns in comparison to others which do not use the column, we're amplifying the magnetic field by 10,000-fold. So this means that we label much, much less because the magnetic force is much stronger. So our beads actually only label about 30% of the epitopes. So you can even go with the same clone antibody on your same cell again and actually put a fluorochrome on it. Sorry. This is just a number of games that we played out at our own R&D where we see that if you wanted to have 5,000 events of CD4 T cells, CD4 positive T cells. These come at about a rate of 0.18% in your tissue. This would mean that you would need to isolate or record about 8 million events. At 2,000 events per second, this normally takes you just over an hour, just for one sample, over an hour. Now, normally you don't have one sample. Let's say you have 20 samples, which is nothing out of the ordinary. It would take you over 22 hours to analyze these cells, to get those 5,000 CD4 positive T cells. If you do the isolation and have these also 5,000 events, so the 20-minute isolation, and do the same thing, getting 5,000 events, all of a sudden it takes you about 20 minutes. It's a huge time saving here, just because you did a 20-minute isolation step, enriching for the cells that you're looking for and getting rid of all the bulk that you're not interested in. Of course, as the cells get, the frequency rises, also, of course, the time difference are reduced, basically. But it's still, even if you're looking at all T cells, it's 2.5 hours versus 16 minutes. So it's a huge difference of time saving when you do the enrichment. This is basically what we're trying to bring across. This is Pfizer, data from Pfizer in the US, where they beta tested our CD45 TIL microbeads human. And what they were looking for, they're working with humanized colon cancer PDX models. So they're looking for the human TILs within these PDX models. And basically, they were isolating them with these TIL beads. And they got very nice, almost no loss in the negative fraction, and a very good isolation or enrichment in their positive fraction of these TILs. So it's really a nice way to get these TILs isolated. As a company, of course, we offer many ways to get to your goal. We have the normal manual isolation, where it works with these stands and magnets and the columns. This is for manual, basically, isolation. We also have the semi-automated one, or semi-high throughput, which is basically a 24-column block. So if you're working with larger mouse cohorts, or if you have multiple samples, you can do 24 separations at the same time. You can also do them fully walk away. So this is the automated version. Then you basically, it does the labeling and the isolation by itself. So the data that was actually produced here was produced on this Automax, this machine over here. And then there's the high-end version, which is basically one of these. So the high throughput put into a TCAN handling station. So there you have 24 samples that are fully hands-off, walk-away instrument. Okay, so just to give you another idea what actually is possible with these different isolation steps, here's just an example. So you're starting off with the dissociated tumor cells. You have use the CD45 TIL isolation. So what happens is all your CD45 beads will be here, and everything else goes through. So now you can take out basically your CD45 TILs and continue with these unlabeled cells. So moving one step further, for example, in this case, they were also interested in the tumor-associated fibroblasts. So we have a kit also for that. So basically, we keep the tumor-associated fibroblasts on the column. So we isolate them afterwards when we take it out of the magnet and work with these cells as we continue on. Or with the last step even, you can then use the tumor cell isolation kit, keeping everything that is non-tumor on the column, which is released afterwards, and your tumor cells going here through. So basically, just in this, it's the same sample, all the same sample, and going this way, you got your TILs, your tumor-associated fibroblasts, and your tumor cells, all in different fractions. So depending on how you plan your procedure or your experiment, you can really have a lot of ways to play around with these to get your target cells. But then there's even the next step. So now this is brand new. It's called release technology, where again, you have your target cells and your non-target cells. You're now putting on a release complex, as we call it, which is an antibody with the fragment dextrin and biotin. So you have a biotin here, which then needs the antibiotin microbead. And basically, this construct here is now your labeled target cell. Again, you go over the column, just as before. Everything that's labeled stays on the column. Everything that's not labeled will go through. But now, moving on to the elution step, where we take it out of the magnet, we add a bead release reagent. This means that in this step where you elute, you right away also cleave off the antibody with the bead, and only the complex here stays on your cell. So in this case, you already have a bead-free cell. So you can take those and right away go for a second isolation. So that would mean you'd have your, let's say, CD4s, and those you can right away put a CD25 marker on it, and you have your Tregs that were in your initial tumor. Had you taken the CD25s right away at the beginning, you would have never identified them because they are such a small target population. But if you want to even go further, it's optional. You can add a release reagent and actually get rid of the complex as well. So in this case, you would have an absolutely labeled-free cell. This takes another 15 minutes. So actually, time-wise, you can stop here and put the next label on it with the next antibody. But if you want it completely free, then you can also do that extra step. So this gives you unlimited possibilities of what kind of subpopulations you want to isolate. Here, just the dot plots. So the original fraction, the isolated fraction CD4 positives, here still with the little tilt because of the biotin complex that is still attached. And this flips over when you release the biotin complex at the end, and then you have your pure CD4 population. The purities are also great, above 80%, and you also can see here the comparison, bulk to isolated tumor. Of course, the increase is enormous because you've basically gotten rid of all this bulk. And then again, as I mentioned, the possibilities, your imagination is the limit because you can release these beads and then afterwards go for any other. So this is the Treg example I just mentioned, but also if you, for example, are interested to go in RNA sequencing afterwards, you basically isolate your T cells, get rid of it, and then use, for example, a dead cell removal kit. Now, dead cell removal kit, if you have more than 50% of dead cells in your sample, which sometimes can happen because tumor samples are quite dirty or maybe with lots of dead cells in it, it will not work properly because it's basically overwhelmed by all these dead cells. So what we did in this step here is that we first did the CD4, CD8 isolation, reaching in there and picking out those T cells, and then use the dead cell removal kit to really clean up your sample. And when you send that to sequencing, you're not only saving time because you're working on only the cells that you're interested in, but you're also saving money because you're not sequencing all these other cells that you're not interested in. You're only focusing on your T cells that you wanted isolated and you wanted to sequence those. So it's really a great way to clean up your sample. These are other possibilities. So for gamma delta T cells, you can use the CD3 TIL beads, for example, and then use the TCR alpha beta biotin microbeads. There's actually data here as well to this. So it's really whatever you're interested in, it just enables you to get to those subpopulations that you weren't able to identify previously. Then, of course, if you go into analysis, your antibodies are a very crucial part of these analyses. And we at Miltenyi, we are actually changing our hybridoma portfolio to recombinant antibodies. This means that we're changing all of them to have these great advantages that recombinant antibodies are. So these are three major reasons. The first one is, since they are recombinantly engineered and generated, they are always the same. They will never have any side chains that appear in hybridoma antibodies. They will always be the same. They're also all using one IgG1 isotope in the backbone, so this means you need one isotype control only for all your antibodies, just one isotype control. And then since we already had the sequence here anyways, we basically messed around with it, and we removed the FC region, so we mutated it. This means it cannot bind to any FC region, meaning you don't have background from that. So basically you have a very reproducible antibody that's always alike, just need one isotope control, and you don't have background with it. And it all comes at the same price as the hybridoma antibodies. So it's really a great advantage. You do have to mention that the full power of this basically comes into play if the entire panel is of reaffinity. Reaffinity is the labeling, the brand basically that we gave it. Because if you have one antibody in there that is not reaffinity, you will still need to block, you will still have some background. So it really comes to full power if you have the entire panel. This is especially true for tumor tissue. This is due to the fact that in the tumor region, we saw that the FC region is actually upregulated slightly. So therefore, you get even more background. So this is without any blocking, comparing the reaffinity to hybridoma. You see a high background here, which is not unexpectedly because it is not blocked. But if you do block it, of course, you get rid of most of the background, but you still have some left. And what I want to point out is that even with blocking, you have a higher background with the hybridoma than without blocking using the reaffinity antibody. So this is really a great advantage to get rid of all this background that you're working with. And now to the last topic. So this is basically now also something brand new that we just came out with. We're now able to use all these FFPE samples that are out there, and we're able to dissociate them into single-cell suspension and then actually sort them. So this is the workflow basically. We need 50 micrometer sections. So usually you would have a 4 to 10 micrometer section, but we actually need 50 micrometers because you want the cells to stay intact, because otherwise you'd be cutting them all open. And then afterwards, the deparaffинation, rehydration, antigen retrieval, it all happens on the tube. which is on this instrument, the Genomax. And then when you have what it does here, basically your single-cell suspension, you can either go for flow sorting and then downstream DNA analysis, for example, or you can actually sort before that. Now, the difficulty with sorting is that due to the fact that you're working with an FFPE sample, so a fixed sample, all your epitopes on the outside will have been degraded. So basically, it's very difficult to find antibodies that still work on this tissue because you basically degrade all your epitopes. But what we were able to find or use are internal ones, cytokeratin and vimentin. So this is the unsorted version. So we have a split here of all the cells. So if you were to analyze this, you'd get a lot of bias in there. You don't know what your read is coming from. But if you sort them, again, you're cleaning up your material. You're actually only concentrating on what you're interested in. And you get a nice positive population of these are tumor cells, these are non-tumor cells. Here just some numbers of how it actually works. And the advantage here is that you have basically, these are just some genes we picked out where you can see it. You can easily discriminate, for example, germline mutations versus somatic mutations. This is something that just is very easily done. And also you increase the sensitivity, as I just mentioned before, because you're really getting rid of either the non-tumor or the tumor cells, depending on what you're looking for. You're really increasing your sensitivity of the NGS downstream. And the great another advantage is that you do not need a matched normal control, which is usually something that might be missing in all these samples that have been stored for decades. There might not be a matched normal control available anymore. But due to the fact that you have these vimentin positive non-tumor cells, they already serve as this control. So basically, you do not need this extra control. This is a great advantage. And with this, I would like to close and just remind you of the topics that I basically addressed during this talk, where we talked about the dissociation, the automated way to do it with the purified enzymes so that you do preserve your epitopes, so that you get better downstream analysis. The isolation with our technology, so the magnetic beads, to target your target cells and purify and enrich for those. And then also the media, which enables you to get stable cell lines. And then also the recombinant antibodies, which remove your background. And the last section, the FFPE sample, which enables you to dissociate and also sort these old samples to get good data out of those as well. And with that, I'd like to thank you for your attention. And if you have any questions, I'd be happy to take them.